Good afternoon, everyone. So, I am Saquib Sarosh. I am working as a research scholar in Saspani Lab, Center for Sustainable Technologies, Indian Institute of Science, Bangalore. Can you load my slide, please? Today I am going to present on use of packed bed biofilm reactors for the application in intensive recirculating aquaculture system. Next slide, please. Next slide. Before dwelling deep into the recirculating aquaculture system, I would like to show some of the stats related with fisheries and aquaculture sector. India stands third in terms of total fish production, and it is second in terms of aquaculture production. So, if we see this graph on the left side, which is documented by Department of Fisheries, Government of India, the total fish production, which is an inland fish production particularly, has been on continuous rise, or there is a steep increase in the fish production in the country from 2000 year onwards, particularly with the inland fish production. So, recirculating aquaculture system is one such component of an inland fisheries sector, and it has the capacity or capability to enhance the fish production, which can meet the food demand and income of millions by multifold. So the focus of my study is recirculating aquaculture system. Basically, in the figure on the right side, it's a conventional recirculating aquaculture system. So what actually a recirculating aquaculture system is, it has a tank where actually the fishes are grown, they are cultivated to meet the food demand. In culture tank, when we grow the fishes, they'll generate a lot of pollutants, particularly in the form of solids, and the major pollutant contributor there is an ammonia, also an organic. So the focus of my study is particularly the ammonia removal from such systems, because an enormous amount of ammonia is generated into the system, and tackling or handling or removing ammonia is the most challenging and critical step here in such kind of systems. And we are making a use of biological reactors for such system, which are packed bed biological film reactors, being very compact. Yeah, next slide, please. Why we are using a packed bed biofilm reactor for removal of ammonia in this system? The major reason is, like, packed bed biofilm reactor have been tested for the effluents from the wastewater plants, and it has been found that they are quite beneficial in use because they are very compact in size. They have robust nitrification process. With biological reactor, we are doing nitrification process for the removal of ammonia with the biological means. So having a compact reactor and the nitrification rate which can be achieved in the packed bed biofilm reactors with the wastewater effluent is as high as 1000 gram per cubic meter a day. So it has been tested with effluents from the wastewater treatment plant, and the ammonia required in the culture tank for fishes, for most of the species of the fishes to survive, is less than 2 mg per liter. So we have chosen this packed bed biofilm reactor because it has not been tested much with RAS. If we see the scenario in context of RAS, the system packed bed biofilm reactors have not been tested for a loading rate beyond 100 gram per cubic meter a day. So use of these kind of biological reactors, which are very efficient, has been missing. So we have tried to intensify the nitrification process in the system in RAS for the ammonia removal using a packed bed biofilm reactor. And in the second stage of the study, this whole study was conducted in 2 trials. In the trial 1, because we are using a biological reactor, so in biological reactors, the microorganisms grow over certain medium. So those are called biological carriers. We have used a specific type of carrier, and in trial 1, the organisms, the biocarriers were inoculated within the reactor itself. By saying that, they were enriched within the reactor, and in the trial 2, there was a specific period of time in which I have used a separate tank and enriched these biocarriers before introducing them into the reactor system. Next slide, please. So since we are conducting the experiment at a lab scale setup, so we have simulated or modeled the effluents from the actual aquaculture plants using some chemicals, and the composition of the effluent is as such as presented in this slide. So the source of ammonia, as in this effluent, chemically prepared effluent, is ammonium chloride, which is the major salt, and then we have some micronutrients as well present in this composition. Next slide, please. So yeah, these are both the trials presented here in this slide. In trial one, like I stated earlier, we have inoculated the biocarriers. The biocarrier used here is Helix-13. These are the plastic medium which we have used as a carrier in the system. It has a very good surface area, close to 955 meters square per cubic meter. So yeah, when in trial one, what we have done, we have introduced these carriers directly into the reactor and added the sludge from ISC wastewater treatment plant into the reactor, and we have just added an ammonium chloride salt and left it just aerating it for 2 days. And once the ammonium concentration in the reactor have reduced to a concentration below 1 mg per liter, we have started it operating at a normal condition. By normal condition, I means we are feeding it through a feed tank. And in the second trial, we have enriched these biocarriers in a separate inoculation tank for a period of over 150 days. After they were enriched, we can see the change in the color. These are the fresh carriers, white one and the brown one, in which biofilms is already grown. So once they were enriched for over a large period, we have transferred them into the bioreactor and then operated it with the normal feed conditions. Next slide, please. So yeah, this is my experimental setup. It has 3 major components particularly. First is culture tank. The red one shown in the left side is the culture tank. And we can see in the 2 trials, the reactor I have shown differently because both the trials experimental setup is same except for the reactor. In the reactor, the carrier's color can be very clearly visible how they differ after enriching and inoculating them within the carriers. So the third main component is the feed tank, and there are series of pumps that we have used, particularly a dosing pump, aeration pump, and a magnetic pump. Magnetic pump is used to maintain a particular upflow velocity into the reactor, by magnetic pump, we are pumping the effluents from the culture tank into the reactor. And the idea of RAS particularly is to reuse or recirculate the maximum water that is present in the culture tank where fishes are actually growing. So in our setup, in this experimental simulated setup, we are recirculating back 94% of the water present in the culture tank, and only 6% water we are adding every day as a fresh water. So yeah. Next slide, please. So to understand the nitrification, we have measured certain parameters into the system setup, and we have regularly monitored the ammonia concentration, nitrite concentration, total nitrogen concentration, and apart from that, pH, DO, conductivity, and other parameters. So ammonia, nitrite, and total nitrogen we have measured in the culture tank, in the feed tank, and in the effluents from the reactor. Yeah, next slide, please. So these are the results and discussion. So like I explained earlier by explaining in RS, the idea is for fishes to survive, we should maintain a minimum ammonia concentration within the feed tank. Because they generate an enormous amount of ammonia, the ammonia as an intake would be as high as 500 mg per liter, and we need to maintain an ammonia concentration of less than 1.5 mg per liter most of the time for most of the species to survive. So that's why we use a biological system for nitrification process, so that ammonia can be reduced to the desired level and fishes can survive in such system. So in this our setup, there are 2 y-axes. The left y-axis, concentration in the feed tank and total nitrogen concentration, and in the right y-axis, the ammonia concentration, nitrite concentration in the culture tank, as well as the ammonia removal percentage and the stocking density corresponding to the ammonia removal percentage. So in our setup, we have particularly throughout the period of study, there were different ALRs to which we have subjected the system to. By ALR, I mean ammonia loading rate, which corresponds to the ammonia concentration into the culture tank. So we have tried it at different ALRs, starting from a lower ALR and increasing in steps and going to the higher ALR in both of the trials. So in trial 1 as well as in trial 2, both the trials, we have throughout the study, we have maintained or achieved an ammonia concentration which was less than 1 mg per liter. I mean, the concentration of ammonia which is desirable to be maintained into the culture tank. And if we see the results from trial 1, the ammonia concentration as an inlet into the feed tank was close to 270 mg per liter, the highest ammonia concentration. So in this study only at 270 mg per liter, we were able to maintain a concentration less than 1 mg per liter into the culture tank. Throughout the period of study, means this was conducted in 3 steps at 100 ALR, then 200, and then 400 gram per cubic meter a day. So at each ALR, the removal efficiency of ammonia was more than 99.75%. In next slide, please. One slide before this. Just this slide before this. One, yeah. So these are the results corresponding to trial 2. In trial 2 also, we were able to maintain removal efficiency more than 99.8%. Trial 2, since we have enriched the biocarriers there for a period of more than 150 days, we have found that it has been beneficial enriching them, because early on, during the early period of study, we were able to achieve a very good nitrification rate, and the ammonia and nitrite, in terms of ammonia and nitrite concentration also, results were slightly better than what it was by inoculating them within the reactor. The results are presented in the graphical form here also in the trial 2 as well. And I mean, at each ALR, we have also calculated a stocking density. By stocking density, I mean the number of fishes that will be available per cubic meter of the volume. And we have, at the highest ALR tested was 600, at which the stocking density, what we have obtained is close to 60. I mean 60 fishes per cubic meter. So, which is in itself an intensive system where the production could be as high as 60 per cubic meter. So, if we scale it up, it can be quite beneficial. Next slide, please. So the same results which I have shown in the graph is tabulated here in the tabular form. The overall in this table, we can see that the ammonia concentration throughout in both the studies has been maintained below 0.6 mg per liter, which was desired, and we have an removal efficiency of more than 99.75%. So next slide, please. So yeah, what can we take away from this study? I mean, we need to meet the demand of food and nutrition. For that we need to enhance the production, particularly if we talk about in terms of fish production. RAS are already there in use. This system we termed it as an intensive recirculating aquaculture system. Here again, I have to state that we have achieved a very good ammonia removal efficiency more than 99.75%. Ammonia concentration at both the trials at each LR was less than 0.6 mg per liter, which means we have a system where we require a very less space. We have a very high stocking density. Ammonia being the challenging step, and it's the most problematic thing, pollutant being present in the system. If we can remove it or achieve it to such a high efficiency, these systems can be a lot more beneficial and profitable, and can be much more intensive for use in our country context, particularly in Indian scenario. Thank you. That's it.